Hey, good morning. Final week, last unit that we're going to cover. We'll probably throw a couple odds and ends in there next week for you guys to kind of, some of the things that maybe we missed or that we need to recover. But for this video, we're going to be kind of talking a little bit more about gene regulation. I'm going to kind of hit probably about 4 or 5 different things throughout this video. Try to just do one and conglomerate everything into that. And again, any questions that you guys have, make sure you guys feel free to ask. So as we get into kind of the whole part about gene regulation, I think we need to go back and first and foremost, probably take a look at the actual molecule of DNA, and for you guys to go back and remember the whole form of DNA versus RNA, the double helix versus a single strand, just the nucleotides, the nitrogen bases, the phosphates, the sugars and stuff, and kind of how they all line up. So again, I think that's probably the first thing that I would ask you guys to do is go back and just to remember the overall structure of DNA replication, DNA, looking at that whole 5' versus 3' end, and just an understanding that basically which end is what, because that's obviously going to play a big part when we start looking at the whole point of replication, which does kind of lead us into the next little part. I want you guys to make sure we're going back and taking a look at DNA replication, the enzymes that are used, that are basically working during that time, as far as what they do, just kind of the overall process. And again, we always go back to the differences between prokaryotic and eukaryotic organisms. And probably just the biggest thing as we start looking at replication is the case that prokaryotic organisms, basically the DNA is found just in the cytosol, where in the eukaryotic organisms it's found inside the nucleus. But again, the whole leading strand versus lagging strand, the Okazaki fragments, this went back to the big drawings that we did in class. As far as how we end up with 2 complementary strands, semi-conservative approach, and end up with the exact same form of DNA so that cell can go on and eventually reproduce. Okay, so again, kind of giving you guys some ideas here, some things to look at as far as the DNA polymerase, the ligases, the topoisomerase, and just kind of what they overall do during that time. So again, DNA molecule, DNA replication, and again, just a little bit about the differences between the DNA and the RNA. And the RNA that we specifically talked about was the mRNA, the rRNA, and then the tRNA. None of those other ones I would really recommend at this point in time you guys having to know. Then we kind of throw in the whole part of protein synthesis. And so when you start looking at replication, obviously it's very important that we're ensuring that every cell inside a eukaryotic organism has the same DNA. Then we start looking at transcription and translation as the building of those proteins, which we can break down transcription and translation. We can talk about what's happening during transcription and translation, and as far as the DNA turning into RNA, and then eventually the RNA turning into proteins. But we also got to kind of take a step back and take a look at the whole regulation of genes and why and how these are turned on and such. So I would ask that we go a couple different ways with this. Okay, one, we start looking at transcription. What is it? DNA is converted into RNA. We start looking at, again, how that all occurs and what direction is that happening. Remember, the strand is being read from 3 to 5, is being built from 5 to 3. We start looking at the overall processes of the initiation, the elongation, the termination, and that molecule is being built. What's actually happening to the DNA with the junk DNA that we find inside of it of the eukaryotic organisms. Remember, prokaryotic organisms don't have that. And just how is that RNA packaged to get ready for being shipped outside the nucleus? And that comes back from the poly A tail. You got the methyl cap that can basically be that is happening during that time. So again, a lot of these terms are going to come up, things that you guys should be able to figure out as far as the template strand, the coding strand, and again, some of the packets we've done in the past, just the overall process of transcription and the overall building of that RNA molecule. Now, remember that eventually that RNA molecule, relatively unstable small molecule, is eventually going to make its way down into the ribosomes, to where you're going to take a look at a translation process happening. And when we go through and take a look at that whole translation process happening, Again, we're taking a look at the two basic parts of the ribosome. We're going to throw in the tRNA during this time and bringing the proper amino acids to that chain. We start looking at the different sites, the A, the P's, and the E sites that are located within the large subunit of the ribosome, and just what it does, and just kind of again that overall process. And again, as you start looking at the RNA, again, relatively small, relatively unstable. Basically, it's the messenger that is optically telling the ribosome which amino acid chain to make. So again, transcription, translation, differences between the DNA and the RNA, some of those special little situations that are occurring that are preparing those molecules for what is going to happen or what is needing to happen. One of the things that we kind of lose sight of a little bit is the RNA molecule being unstable. Why is it unstable? Why is it important that the RNA molecule is unstable compared to the DNA? And at the end, it's so that it actually will degrade it or breaks down very, very easily. So we just don't have all these RNA molecules that just continues to build proteins. And so we have the ability to turn those proteins on and off. And we don't want those mRNA molecules just floating around, just keep on building proteins that really aren't needed. Because again, one of those ultimate themes that we start looking at with our bodies is efficiency and producing as much as we possibly can, doing as much as we possibly can, but with a low amount of energy that we need to be utilized. So when we go through and take a look at, again, here's your translation notes as we go through, but we go through and take a look at that whole process of gene regulation. There's two different models that we looked at. We looked at the TRP versus the lac operon. Again, I would ask that you guys go through and take a look at the TRP operon, take a look at which dealt primarily with tryptophan, and then we have the lac operon, again, basically primarily in bacterial organisms. Now, the reason why we focus so much on bacterial organisms or prokaryotic organisms gene regulation is because they're easy to see. The eukaryotic organisms are much more complex, so we kind of use these models as a way for us to go through and understand what's happening in ours, but ours being much more complex in that process. But again, when we go through and take a look, it's all about either turning a protein on or turning a gene on or turning a gene off based on either we have enough of a substance that we don't need to make anymore, or we start looking at the process of we have something that we need to break down. So we have too much of something, we need to eventually break that down, and eventually we need a gene that is turned on for that. So again, the TRP versus lac operon, you guys can go into a little bit more specific. Now, we started looking at eukaryotic gene regulation. With the eukaryotic gene regulation, again, much more complex. We kind of start looking at things that our body, we can do in order to regulate whether genes are turned on or turned off. And we've given you here a list of 6 different things that you guys can go through. And some of them are dealing with transcription. Some of them just deal with translation, the building of those molecules. We start looking at, the chromatin modification. One, we can modify the chromatin. We can actually condense it, or we can expand it in order for those genes to be a little more easily accessible, and then eventually go and be read and eventually making those proteins as an end result. Transcription, pretty simple. RNA processing, again, the building of the RNA. Now, when we start looking at the whole RNA processing, we start looking at a little bit more specifically of splicing out all the junk DNA, the methyl caps at the end, the poly A tails at the end, making it a molecule that can eventually work itself to the ribosome in order to be read and make that protein. mRNA degradation, we already talked about why it's important that it degrades. We don't want all these molecules simply floating around and continuously producing more and more and more. We'd be wasting a lot of energy. So therefore, it degrades as soon as that ribosome utilizes it. And then if we need more of that mRNA, again, we just keep building it at the nucleus level as far as eukaryotic organisms go. Translation, again, just the overall steps of translation and how basically we're building that protein based on that mRNA sequence that's coming from the nucleus. And then again, just the overall proteins being produced in degradation and the whole kind of big picture of the central dogma of genetics, if you want to call it that, and eventually our ability to create those proteins, but also break them down if they are not needed. Okay. I think the other part that we start focusing on in here is we start looking at mutations. In the case of what mutations can do for our body. And a lot of times these mutations go unnoticed because of just a simple little substitution, one of those codons, to where it won't have an impact on the building of that protein. But in some of these other cases, if you start seeing the frame shift or you start seeing a substitution to where it ultimately changes the codon into basically producing a different amino acid, well, obviously they can have a different different property. Give the actual molecule, give the actual amino acid chain a little bit different property based on the R group that amino acid is bringing in. So a lot of different severities here, from basically nothing happening with the silent substitution to eventually the frame shift, we can actually shift that whole shift that whole frame over of the RNA molecule until we get a completely different amino acid sequence and a completely different protein. So again, mutations obviously play a big role in this case. And mutations we always generally think of as bad, not always bad. Sometimes they can be positive. In the case of mutations in bacteria, which allows them to be antibiotic resistance, and so there's obviously some good and some bad that come along with those mutations as we start going forth. So tried to kind of tie in a lot today, and I didn't probably go as quite in depth as I think maybe we should have in these videos. But again, anything that you guys need from me, make sure you ask. I kind of focus more on the big picture items as far as just the overall, the protein synthesis and the ways that we can turn those genes on and off, and the bigger picture behind, rather than really focusing on those details. So I would ask that you guys do go forth and look a little bit closer at those notes, go a little bit. Take a little bit closer look at some of those review questions, and just, again, that general understanding of what is happening during those times. Hope everything is going great. Again, feel free to ask any questions, either via email or, again, ask my phone number. I'll be happy to text, call with you guys at any point in time. You guys have a good day.